The views expressed on Skeptically Speaking are not necessarily the views of this station, its affiliates, sponsors, or advertisers.
Bring it on, the skeptics await your letter. Your call, tweet, homing, pigeon, whatever. Desiree Shell gonna raise a little hell, metaphorically that is. Cause we're skeptically speaking.
Hello and welcome to Skeptically Speaking. I'm your host, Desiree Shell. This is the second annual World Changing Ideas episode, produced in cooperation with Scientific American magazine. 10 World Changing Ideas is the title of the feature story in the December issue of Scientific American, and it profiles 10 examples of cutting-edge research and technology that could shape the future of society, our planet, and even our survival as a species. Before we get into details, we wanted to talk a bit about the report as a whole. Mark Fischetti is a senior editor at Scientific American who assigned and edited all of the articles in the World Changing Ideas report. He also oversees the magazine's coverage of energy, environment, and sustainability issues. Welcome, Mark.
Hi, thanks for having me.
So what qualifies a technology as world-changing for the purposes of this report?
So world changing, yeah, it's a pretty broad scope, but we've really focused on ideas that are not sort of just pie in the sky ideas, but in fact are ideas that have been worked on enough so that there's a prototype or something actually physically done that you can test and try, and so you can see that it works, and you can also see that, yeah, in fact, this could really be scaled up to a great product or service that could really change people's lives sort of day to day.
So how did you choose them? Because there's only 10.
Right. Well, we have more than 20 scientists and engineers, all top scientists and engineers in all sorts of disciplines, who are advisors to the magazine. So we begin by polling all of them and saying, hey, in your area, what do you see emerging that could really be significant, have practical implications? So we get suggestions from them, the editors at the magazine. There's about a dozen of us. We are also looking at different disciplines, and so we've got our own ideas of what's emerging. And so we put all that together and then sort of take a vote and do a little testing too, because again, these ideas are things that have to be prototyped enough to know that they could actually work on a larger scale. And that's kind of the key factor that separates these from other sort of fanciful ideas about the future.
Right, and you do go out of your way to say that these are potentially world-changing ideas, right? Like all of these ideas do have a way to go at this point.
Oh, sure. And what's really fun to read about when they're at this stage is you see something being tested or tried, and you think, wow, that's really kind of cool. But then immediately you imagine, okay, but what would still have to be done to really make this part of everyday life? And that's, a great challenge, and sometimes it works and sometimes it doesn't.
Well, let's give people an example, starting with the Ultimate Sustainability Index. What's that?
Yeah, so, there's a lot of people are aware now about the impact of the products that they're buying. So, if you're buying shampoo or cereal or a car or anything in between, what impact does the manufacturing of that product have on the environment? And I think a lot of folks are starting to take that kind of consideration into their buying decisions. But how do you know? How do you know what effect making that shampoo or that car has on the environment? There have been different kinds of studies done about certain aspects of products, like how much carbon dioxide is released during the manufacturing of this product, or how much waste is created during the manufacturing of this product. But there's no real way to tell kind of the overall footprint of this whole supply chain that leads to this thing that you can actually buy. So a number of leading universities and nonprofit organizations and about 80 international companies have all gotten together to form something called the Sustainability Consortium. And they're all working to try to do just that, to give consumers a specific way of checking on the environmental footprint of all kinds of products out there.
Well, and what kind of data would the index monitor then?
You may have heard of this concept called cradle to grave or life cycle. It's everything that's involved in making a product. So let's just take, for example, a magazine, let's say. So you've got paper that comes from trees, and you've got chemicals that are processed to create inks, and then all that gets put together on a printing press, which requires energy. That all gets bound together, and then it gets mailed all over the place, which is transportation. So, paper effect on forests, chemicals effect on the environment, wastewater from the processing, energy usage and efficiency when it's being printed. All those kinds of factors come into play in creating these so-called overall cradle-to-grave life cycle, footprint of this magazine.
It was also mentioned in the article that labor practices would be monitored.
Yeah, that's a sort of this social costs of a product. On and off, there are news stories about manufacturing over in some other country somewhere where the conditions are terrible, or pay and wage structures that are inequitable. So lots of people are starting to consider more of that social aspect of the supply chain. So that's another factor that the Sustainability Consortium is taking into consideration when they're rating how the products are affecting the larger world.
And now the problem isn't that the sustainability information doesn't exist now, it's that there's too much of it?
Well, yeah, that's, as I was saying, there's sort of a number of different indexes that look at discrete stages of the supply chain. So let's say, landfill waste, right? How much material is wasted and ends up in a landfill, or energy usage, right, by a particular factory or supply chain. But they tend to be isolated on the one factor. So the Sustainability Consortium is trying to bring all of that together into a single metric, probably, a number and an icon, or, a term that they're going to come up with, so that when you're in the store, if you think about seeing this number or icon on a package, that gives you sort of the overall environmental impact and social cost. That's the level of simplicity that a consumer needs, and the level of sort of inclusivity of all the factors that could really end up being a single index that everybody can use.
So beyond consumer use, there are ways that this index is also going to be used.
Why are 80 international companies interested in this? I mean, part of it's altruism, part of it's image, part of it's there are practical benefits. So, if you can reduce the amount of wasted material in making something, then you're probably reducing your costs, right? If you can use energy that's more efficient, you can reduce costs. If you can use less water, it's maybe less cost, but it's also less processing. Wastewater has to get processed and then it has to be sent somewhere. So it's kind of a big practical problem for a lot of manufacturing companies. You can do good for people, do good for the environment, and do good for the company all in the same way. And so what's going to happen is, or what's happening now actually, is the companies are looking at various stages in the supply chain to find out where different sorts of wastes or inefficiencies are and how they can be improved, which then benefits everyone involved.
Okay, well, let's talk about the consumer aspect of this again. This kind of thing does exist other places, as you were saying. How do we know that this information will be accurate for the consumer? Because is this all self-reported?
Yeah, I guess it would be self-reported. Some of this data is public. And so like wastewater, let's take, for example, it's all regulated, and so it has to meet certain requirements, and then that sort of thing is monitored. Landfill too, when waste is taken from a manufacturing plant and it's dumped somewhere, I mean that's sort of monitored too. So some of it is public, but some of it's private, and you are trusting the group to some degree. But I guess their point of view is that if they're all working together, they're kind of monitoring each other too. These are not companies that are cooperating; they're all different sorts of companies. So there's Dell, there's Walmart, there's different companies that are not in competition with each other. So I think that's probably the best you can do. I don't know how else you could come up with an index that's so inclusive and yet somehow is all based on information that everybody has access to.
Well, so how soon will we actually see this in consumer use then?
Walmart is probably at the forefront of this right now. They've created scorecards that, for their own buyers, they have about 400 buyers who procure products. And so the buyers are using this scorecard to assess how well their suppliers are doing in reducing environmental impacts. So that's begun, and the consortium's hoping that by 2015, all of this will be in place and will be up and running. And so that's when the final step, which is, some sort of number or icon or something that would be available to consumers to check out.
There's a lot of discussion about sustainability, so let's see if people put their money where their mouth is.
That's an interesting part of this. I'm glad you brought that up because there's this sort of sense that, environmentally beneficial, well, benign products are going to, quote, cost more. And so consumers are kind of interested, but they're kind of wary, I think, to some degree. So we'll see. You're right.
Mark, thanks very much for being here.
Sure. My pleasure.
I'm speaking with Catherine Harmon, an editor at Scientific American magazine, where she covers health, medicine, and life sciences. Her first book is about octopuses, and it will be coming out this fall from Current, and we will be calling her back at that point to get her back on the show. Good to have you here, Catherine.
Great. Thanks so much for having me.
So tell us about this new breathing foam.
Well, it's a foam, and it actually doesn't look, I'm told, unlike shaving cream. But unlike shaving cream, this foam is full of tiny microbubbles of oxygen. And the idea is that you can just inject it straight into the bloodstream without having to help somebody who's having difficulty breathing because their lungs are injured, for example.
So how does it work? What does it do?
So because it's full of tiny little microbubbles of oxygen, once it's injected into the bloodstream, our hemoglobin, which loves oxygen, thankfully, absorbs that oxygen right away and distributes it throughout the body. So for many patients who, sustain a critical injury, maybe have a heart attack or their airway is blocked for some reason, their lungs simply aren't working, to get oxygen to the rest of the body. So this is a way to totally bypass that and help oxygenate the body, at least temporarily.
So now why doesn't this cause an air embolism? Because my assumption was that when you inject air into the body, that's the result.
And that has been the problem for a long time. Back, over a century ago, people knew that it was a problem when we couldn't get oxygen into our bodies. So they would just, inject oxygen straight into the blood. And that is very bad news. As you mentioned, it causes the embolisms, which, can block blood flow and cause a heart attack or stroke. So because these bubbles are so small, they're just about 4 microns wide, and because the oxygen gets absorbed so quickly, the way they're designed, they collapse almost automatically to a very tiny, almost disc-like shape, which isn't big enough, fortunately, to cause these problems.
And now they use sound waves to cause the ingredients in this to self-assemble into microparticles. What does that mean?
It's a pretty complicated process, and some of it's probably beyond me a little bit. But so what they do is the basis for these microbubbles are lipids, fats. So they send a solution of these lipids into a super oxygenated chamber, and then they blast that with sound waves. And they've discovered a way to allow that to turn into these tiny little microbubbles that are just the right size to hold enough oxygen, but also to be small enough to collapse and not cause any trouble in the bloodstream.
And microparticles with lipid membranes are actually already used in other applications, correct?
Exactly. Yes, we already use them to deliver drugs like chemotherapy, although in those instances they're designed to release the drugs very slowly. But in this case, since we need to get oxygen to the body quickly, these are designed to just be kind of instantaneously dissolvable.
So now how much of an improvement is this? Like, how do paramedics usually deal with someone when they stop breathing?
Well, the first thing they can do is apply an oxygen mask. You'll see those just over the nose and mouth to kind of supply oxygen to people who are having a little bit trouble breathing or getting enough oxygen. But as you can imagine, if the lungs aren't really working or if something's blocking the airway, that's not going to do a lot of good. So what they can do is they can insert a tube down into the throat, but that's kind of a complicated process and can take some time. And if a patient's very injured, sometimes they just can't do it in time.
We should probably point out that the lack of oxygen causes the brain to start to shut down. So every minute counts at this point.
Exactly. So really you only have, a few minutes until there starts to be potential danger to the brain for severe brain damage. And then after that, cardiac arrest can happen later. So yes, you need air as soon as possible.
Now, how's the foam being tested at this point?
So right now it hasn't gone through any human trials yet. It's been tested on rabbits in the lab, and they've found so far that it's been able to keep rabbits alive for up to 15 minutes after they've had their windpipes blocked. So for 15 minutes, these rabbits don't take a single breath, but just with this oxygen foam being injected, they seem to be okay.
Now, are there any concerns?
Sure, there are some concerns, which is why it's not already being used in ambulances.
Right.
Because it's absorbed so quickly, we need to keep injecting it into patients so that they can have a constant supply of oxygen. With that, however, it's currently giving a little bit too much liquid because the bubbles are dissolved in a saline solution. So if you can imagine if somebody was hooked up to this on an IV, they would, after maybe 5 or 10, 15, 20 minutes, just have too much liquid in their body, and which can also be very dangerous.
Another concern is that without normal respiration, carbon dioxide builds up.
Exactly, yeah, which is another danger because, yes, when you're exhaling, you're getting rid of this, this toxic carbon dioxide. And if you're not breathing at all, then you're also not getting rid of that substance.
Really, the total lack of oxygen, though, is probably worse, right?
Yes, yes. The doctors I've spoken with said, well, your body can handle a little bit of extra carbon dioxide, but it's oxygen that we really need.
So you mentioned, we haven't started seeing this in use in ambulances. When might we start to see that?
Well, that's still a question. It sounds like there's some challenges in designing this sort of trial because its use is for these very intense emergency situations where you're really looking at life or death or at least brain damage, and it's a crucial matter of moments. But to participate in a clinical trial, generally the patient needs to give their informed consent, which usually isn't going to happen in this situation. So I think they're working at ways to make this a little bit more easy to administer so it doesn't require so much liquid, and also trying to figure out the best way to test it in humans.
So it's still a while then?
It might still be a while, yes. But ideally, it could be in ambulances, it could be in intensive care units where maybe they might need it just for those extra few minutes it takes to get somebody, say, on a heart-lung machine, or give them a breathing tube. In the battlefield, it could also be useful. Or, maybe even just in CPR kits where somebody just needs a few extra minutes worth of oxygen.
Catherine, thanks very much for being here.
Sure, thank you so much for having me.
And we'll be right back with more of Scientific American's world-changing ideas on Skeptically Speaking after this.
Skeptically Speaking wants you to question everything. To be a part of the discussion, call and record your question on our listener question line at Speak 73101. That's 773-257-3101. Long-distance charges may apply. Sunday nights at 6 Mountain Time, join our Ustream chat room to listen and ask questions as we record interviews live. Just click the Listen Online button at skepticallyspeaking.com. You can also email your questions and comments to live@skepticallyspeaking.com, or if you're on Twitter, tweet us at Skeptical Radio. To find out where Skeptically Speaking airs near you, to listen to past episodes, or download the weekly podcast, check out our website at skepticallyspeaking.com. While you're there, follow the links to connect with us on our Facebook page, our Twitter feed, or to subscribe to the show in iTunes. And now, back to the show.
Welcome back to Skeptically Speaking. I'm your host, Desiree Schell. We're talking about cutting-edge technology with the writers and editors of Scientific American's annual World-Changing Ideas feature. I'm here with David Biello, Associate Editor for Environment and Energy at Scientific American. He's currently working on a book about whether the planet is entering a new geologic age caused by humans, and if so, what we should do about it. Good to have you here, David.
Thanks for having me.
So tell us about using soybean oil to clean wastewater.
Well, these are called by scientists directional solvents. And essentially what that means is you can choose what you want to put into solution and what you want to take out. So the problem they're trying to solve is the fact that down here in the United States, we produce some 2.5 billion gallons of contaminated water every day just in the oil and gas industry. And it would be awfully nice if we could do something with that water. But trying to treat it by boiling it has proved too expensive, too energy expensive, and too cash expensive. And there aren't really any other good solutions. So a long time ago, or a few years ago, I should say, a graduate student by the name of Anurag Bajpayee was working actually on cryopreservation. This is freezing yourself and putting yourself in the freezer and keeping yourself ready for I guess the next go round. And in that cryopreservation, they use these what are now called directional solvents to keep the cells from getting frostbite. if you're going to freeze your brain, you don't want your brain to get frostbite. So he was discussing this with some of his thesis advisors, and they said, hey, would that work for water treatment? Because, of course, treating dirty water, as I just mentioned, is a big, big problem, because in addition to oil and gas water, there's all kinds of other water that we'd like to treat, including the oceans. So we could turn all that seawater into fresh water that we might be able to use. So he said, well, I don't know, but let's find out. And they tried a couple different things. Soybean oil was the first one that worked effectively. The problem with soybean oil is that you need about a swimming pool's worth of soybean oil to get a cup of clean water. So that's not a very effective method. So using chemistry and finding some things that are like soybean oil, they found some more effective solvents. And assuming that this can be scaled up and turned into an industrial process, it could really change the way we deal with all this dirty water that we currently essentially lose.
Now, this is one of the few entries in the World Changing Ideas series that doesn't actually rely on new technology, right?
That's right. There's nothing super new about this. In fact, one of the chemists I spoke to said, this is really cool. And what's most amazing about it is that there's no reason this couldn't have been discovered 100 years ago. It just wasn't. Or if it was, nobody remembered it or recorded it. Now, there are many hurdles still to be kind of proven out, like the soybean oil example. These directional solvents still aren't the most efficient at removing water from its contaminants, and that's what these do. I should probably explain that. Rather than you think of a typical solvent, you're taking the impurity out of what you want to make pure. In this case, you're actually taking the water out of the impurities. So the soybean oil, or whatever directional solvent you're going to use, bonds to the water, pulls it out, and leaves all the contaminants behind, whether that be salt or whatever else. So it's kind of the reverse of what we normally think of as a solvent. But it is certainly, like I said, could have been discovered a century ago.
Well, now you mentioned sort of the scaling of this for large amounts of wastewater, because right now it's only being done in small batches.
That's right. And that's going to be a big challenge. So they have to figure out, first of all, the best solvents, and then they have to build a process that can do this kind of both easily, and that seems to be the easy part, funnily enough, but also quickly. So currently the way they do it is they allow the directional solvent to work, and then the water kind of drips out under gravity. But that takes a long time, and if you want to process 2.5 billion gallons of water a day, you don't have a lot of time to waste. So they need to figure out a way to speed that up with pumps and whatnot, and that's what they're working on now, along with the chemistry work to try to find even better directional solvents.
Well, now, you did mention desalinization, and am I incorrect in thinking that is actually almost more important than cleaning wastewater?
It's definitely, in the long term, more important, because there are more and more of us, and we all need fresh water, not in addition for just our own drinking purposes, but also for agriculture and all these other things that we like and rely on. The problem there is that in seawater, as salty as it is, there's not that much contaminant. The water is pretty pure, so it's difficult for these directional solvents to work on seawater. That's not to say it isn't possible. It just is going to require even more development. These directional solvents work best on really, really dirty water. And that's a good thing in its own right, because we have a lot of that too.
So really, for a number of reasons, there's a lot riding on this world-changing idea.
Absolutely. And it would put an end to essentially the number one cause of man-made earthquakes. It would stop us from wasting this water. Essentially what we do with it is pump it back underground and lose it for all practical purposes forever. So that's 2.5 billion gallons of water that are going away every day. And in a world that's facing increasing water shortages, that is definitely not a good thing.
David, thank you very much. Wonderful to have you here.
Thank you.
I'm here with Daisy Uhas, a science journalist who's written for a number of publications, including Scientific American and Scientific American Mind. Welcome, Daisy.
Hi, thank you.
So you're here to talk about a new way to do prenatal genetic screening for single gene disorders. But let's just start out by explaining what a single gene disorder is.
Sure. So there are actually about 3,500 different single gene disorders that result from just one mutation or one problem within one gene. And so, for example, Tay-Sachs or cystic fibrosis are caused by a problem within one gene.
And how are prenatal screenings for those kinds of disorders usually done?
There are a number of options out there currently, but many of them are invasive. So that includes chorionic villus sampling and amniocentesis. And these methods basically require taking a sample from either, say, the amniotic fluid or placental tissue, and they carry, as a result, a slight risk to pregnancy, which deters many women from taking them. There are a few screening tests that have now become available for chromosome-level disorders, so things like Down syndrome. But what's really revolutionary or really different about the idea of full genome sequencing done non-invasively is you could get the complete picture, you could get everything within one test that carries no risk to the pregnancy, unlike chorionic villus sampling and amniocentesis, these older methods.
So how exactly have they figured out how to access a fetus's full genome through the mother's blood?
Right, so this is actually really amazing. In the 90s, researchers found out that a portion of the cell-free DNA in a mother's blood, an expectant woman's blood, actually came from her fetus. Since then, researchers have been working on distinguishing and separating then the fetal DNA from the maternal DNA. In the past year, there were 2 papers that came out, and what they basically demonstrated was that if you can deduce mathematically the ratio of maternal to fetal DNA, you can separate the two and reconstruct, once you have those separate sort of chunks of DNA, reconstruct the full genome from that. So, for example, in those 2 papers that came out, one looked at just a sample of blood from the mother and was able to separate mother and sort of that DNA which did not come from the maternal genome. And as a result, that was therefore fetal, and they could build up the whole genome from there. And then in another paper, they actually used a sample from the mother's DNA and the father's DNA. They took a saliva sample. And from those 2 sort of maps of genes, they could separate what came from mom and what came from dad, and then what was unique to the fetus, because there'll also be mutations that occur uniquely in the fetus.
So now this means the possibility of non-invasive testing then?
Exactly. And that's actually a great new option for many women who don't want to risk their pregnancy. It also means that you don't need a skilled obstetrician or a skilled practitioner to carry out a procedure like chorionic villus sampling or amniocentesis. And that means you could reach more women than ever before, provided the cost of sequencing goes down.
Well, now you just brought up one criticism. So yes, the cost is one.
It is quite expensive. It is something that every year these technologies for sequencing are improving. They're becoming more accurate, and they're becoming cheaper. And so this is sort of an option that's riding that wave, I guess. As that cost goes down, this becomes more and more feasible, and it's clear that this is improving and the costs are going down. So it should become more possible, but that is actually a real practical limitation now, is the cost.
Well, and the technology, it seems, only recently became practical at all.
I think that's correct. It is, however, improving rapidly, which is why this is clearly, I mean, what the papers demonstrated this year was that on a technical side, this is totally possible. There are other obstacles, but from the geneticist perspective, providing a full genome of a fetus is doable and can be done without touching the fetus, which is amazing.
Well, and another criticism of this is the accuracy, because
Right.
This is definitely a sensitive area. The potential ramifications of a false positive for something like, I'm thinking, Tay-Sachs, could understandably cause a parent to decide to terminate a pregnancy. And even if that wasn't the direction they wanted to go, that's a huge amount of stress for a new parent in trying to
Right.
plan for parenting a special needs child.
Right. And you're touching on actually, I think, several really important points there. In terms of the accuracy, there's always concern there. I mean, even with a perfect picture, there is still a lot of uncertainty about how to interpret and how to counsel parents who then receive this genome. It's challenging already for, say, an adult to receive their full genetic blueprint and then be left with questions about what does this mean, or if I have susceptibilities for this or that, what would that mean to me? Helping parents then is also hugely challenging because these are people who are trying to make informed decisions and trying to prepare for raising their children. There's definitely a lot of concern about how do you interpret this much information. And I think that's something that remains actually a bigger challenge than the technical issues of creating a really accurate blueprint. Being able to counsel accurately and explain what we can and can't know from this information is huge.
And that's actually something that you touched on in your article. The idea of interpreting the genome at all is an obstacle.
Right. It's amazing. We still really don't know how to interpret most of the variation we see. And so how do you tell someone? I mean, it's hard to imagine what to do with this information when you see strange variations or strange mutations that we just don't know what they mean. And there is, I think, a fear that people might act too quickly when they see that, or might panic and say there's something wrong with my child, when in fact, I think what should come out of this at some point is the realization that none of us have a perfect genome. None of us are that perfect. We actually all carry all kinds of weird mutations and weird quirks, and all of us carry real risks. So figuring out how to talk to parents about this is a big deal and is a huge challenge.
And again, this isn't a problem that's specific to prenatal testing. Scientists who are researching anything to do with the full genome at all are facing the same kind of issues.
Right, absolutely. I think, yes.
And this is very much a developing field, people have to remember. There's a lot of gray area.
That's right. Yes, definitely. And it's definitely, I think, increasingly clear that geneticists and doctors and counselors have to work together and try to sort of improve their own literacy and the literacy of sort of the general public in terms of what this means and what we do and do not know.
Daisy, thanks for your time.
Thank you.
I'm talking with John Carey, freelance writer and former senior correspondent from Business Week, where he covered science, technology, medicine, and the environment. His recent stories have appeared in publications such as Yale Environment 360, Conservation, Scientific American, Science Business, National Wildlife, and Yahoo News. Good to have you here, John.
Great to be on the show, Desiree.
So, typically when we think of drones, there aren't many positive connotations, really.
Yeah, we really think of those great big predators that are flying around Afghanistan, killing those bad people. And of course, that's been very controversial. The government's also using those big drones like predators to fly over the borders and look for people coming in.
Now, how do drones work?
Many of us, including me, I don't know about you, played with radio-controlled airplanes when we were kids. And essentially, a drone is just like a big radio-controlled airplane, except the key difference is that it's autonomous. You can tell it where to go ahead of time, and it has a GPS unit and other guidance equipment so that it can actually fly a route that you have pre-programmed into it.
So now scientists are actually starting to kind of hack these together for themselves, correct? To help with their work?
Yeah, that's been a very interesting phenomenon because in many cases it's very, very difficult to, survey a huge amount of rainforest to look for orangutans or to count stellar sea lions in Alaska, because if you drive up in a boat, they all dive into the water, and if you fly in a plane, they all dive into the water. So how can you possibly get close to these animals and cover a lot of territory in a way that's sort of very quiet and unobtrusive and yet sophisticated enough to do the kind of scientific work that people want to do? And so the scientists have figured out that they can either buy some of these systems from companies like Air Environment, which makes the very small military drones, or can they actually build them themselves?
Now, how much would something like this cost?
Well, if you go to the commercial vendors, you can buy a very sophisticated drone from a company called Gateway for about $65,000. If you go to Air Environment, which has been this pioneer, they've done a whole bunch of things that you've heard of, including some of those solar-powered airplanes. You can get a system that includes 3 airplanes and all the ground support gear for about $60,000, so $20,000 per plane. If you go online and buy a little hardware, almost like a circuit board with the right circuitry GPS units on it for about $50, and then buy a radio control plane off the shelf and put all those things together, you can make your own for probably less than $2,000.
Okay, so this is not only effective in terms of process, it's cost effective.
It's very cost effective, and the cost is coming down. And in fact, what we're seeing now is this almost explosion of interest in what you might call the do-it-yourself community. In fact, there's a whole semi-commercial business, DYIDrones.com, that will sell you some of the base equipment and then provides a community of these kind of hobbyists that can help you build your own.
So now, some interesting things are being done, but what does the future of drones look like then?
Well, that's a very interesting question, and all the people I talked to, well, the real applications are things that we probably can't think of yet. But we can think of a lot of things, and people are already doing a lot of things. You think of farming. Farmer can go out there and fly over the fields with one of these drones that has a GPS unit that can actually then look down on the field and to look at things like the amount of moisture using sensors in the drone, so he knows exactly where to where to irrigate, or he can look at how the plants are growing and know exactly where to fertilize. So, or oil companies can fly along pipelines searching for gas leaks or oil leaks or something like that, and so you can look for oil spills. One huge market that the companies are looking for is the whole law enforcement community, where they imagine every one of the 18,000 local enforcement agencies would have a little fleet of drones that they could use to chase bad guys. Instead of having a car chase, you could just fly along and follow them wherever they went.
Well, and now one of the problems is that the Federal Aviation Administration has actually banned commercial uses of drones, right?
Yeah, this has been a sort of a block on a lot of these potential uses because, when you think about it, there are something like 100,000 commercial flights a day, planes flying through the air, and there's up to more than 200,000 small private planes, general aviation. And imagine suddenly if there were thousands of drones that were going up in the air too, what kind of chaos there might be. And so for now, the FAA has said that these drones, except for some of the government use, like we talked about earlier, the Border Patrol, they have to stay below 400 feet and they can't go very far. So that is sort of slowing the whole commercial uses down, but it's giving people time to think about what those commercial uses might be, and sort of what we really want these drones to be used for before it becomes possible to actually do it. Now, the FAA, a law was passed this year requiring the FAA to come up with regulations that would allow commercial uses. And so that's in progress right now. And the earliest those regulations might be finalized would be in about 2015.
Well, there's some potential ethical issues, I would assume, with privacy and such. So how is that going to be dealt with?
That's an interesting question. I think that's still one of the big unanswered questions, because you really could go up there and fly around and spy in people's backyards or follow people around unobtrusively, do all sorts of things. If sort of environmental activists want to go fly around a company looking for leaks, is that an invasion of privacy? And all of this stuff is going to have to be worked out.
Fantastic. John, thanks for being here.
My pleasure.
And we'll be right back with more world-changing ideas from the staff of Scientific American after this.
On the next episode of Skeptically Speaking, we're looking at debates over the ethics of medicine and medical research and the future of new medical technology. We'll talk bioethics and public policy with Center for Inquiry CEO Ronald Lindsay. And we're joined by Dr. Rob Tarswell to discuss nootropics, the so-called smart drugs that might make designer brains a reality. That's next week on Skeptically Speaking, on your local radio station, or online at skepticallyspeaking.com.
Welcome back to Skeptically Speaking. I'm Desiree Schell. This week, we're taking a closer look at some world-changing ideas as chosen by the writers and editors at Scientific American magazine. Here to talk about our next world-changing idea is Ferris Jaber, an associate editor at Scientific American and a freelance writer based in New York. Welcome, Ferris.
Hi, thanks for having me.
So you start out your World-Changing Ideas article claiming that DNA is passé. Bold
Yeah.
words, sir. So, how so?
DNA is probably the most important biological molecule on the planet. Every single life form on this planet uses DNA to both store and to transmit hereditary information. So everything from viruses, if you consider them life forms, to nematodes, to jellyfish, reptiles, fish, birds, mammals, humans, everyone uses DNA. But there's no real reason why it should be the only molecule that can store and transmit hereditary information. And so, using that premise, scientists have been for quite a while tinkering with both DNA and RNA, actually, seeing can they make variations of these molecules that can do the same thing or even store and transmit information in better ways.
So tell us about what they've found, which is XNAs?
So XNAs refer to a specific class of 6 new molecules. It stands for xenonucleic acid, xeno meaning foreign. And so XNAs can do everything that DNA can do, and they have the same nucleobases as DNA. So DNA has these 4 nucleobases, the famous A, G, T, C letters, and XNAs have the same ones, but the molecules that make up the backbones of XNAs are completely different from the molecules that make up the backbones of DNA and RNA. So DNA stands for deoxyribonucleic acid. The deoxyribose in that name is the sugar molecule that's in the backbone. XNAs do not have deoxyribose or ribose. They have completely different sugars and other molecules in their backbones.
And so they also then created enzymes that work with the XNAs.
The XNAs alone, or even just DNA or RNA alone, they can't really do that much. They have the information stored within the patterns of repeating nucleobases, but if you don't have the enzymes to work with these molecules, you can't replicate them, you won't have evolution, you won't have the ability to pass on traits to your offspring.
Now, this is the really interesting part. So XNAs can evolve.
Yes.
Explain that to me, please.
You have the XNA molecule, and it's got its repeating patterns of nucleobases, and you want it to make copies of itself, and then you want it to transmit any errors or changes that happen during that copying process. Because that's a big part of how evolution happens is, mistakes happen spontaneously, and those mistakes are faithfully recorded, and sometimes those mistakes lead to advantages. And when they do, you have new traits evolving. So in order for this to happen with XNAs, you have to sort of put them in a solution with their collaborative enzymes, and you have to see if when they make copies of themselves, are they faithfully carrying along any of the changes that happen? And XNAs are able to do that. And there's a specific example that was discussed in the study where they designed a particular type of XNA called HNA, and the H is just because of the particular molecule that's in its backbone. And they sort of threw a bunch of HNA at this particular protein, and they wanted to see how well the HNA would bind to that protein. And most of the HNAs did not do a very good job, but a few did a better job than the other ones. So then they took those few that did a better job, and then they threw those at the protein again, and they kept doing that over and over again. And eventually they had HNAs that did a pretty good job at binding to the protein. So that was their example of how this XNA could evolve, could change one of its abilities and then faithfully transmit those changes.
Then that means all of this means that it appears that this makes it possible to create life forms in the lab, ones that don't depend on DNA.
That's right. So if all known life on this planet depends on DNA and RNA and the enzymes that work with those molecules, and if you can create an alternative system, then you can create a life form that uses that alternative system instead of using DNA and RNA. So now it's important to note that XNAs cannot work 100% without DNA. They still kind of need DNA to hold their hands a bit in the replication process. You can't really make lots of copies of XNA unless you have a bit of DNA to work with, because the enzymes they created, they translate XNA into DNA and vice versa.
Right.
It's definitely not a huge leap to sort of create a 100% synthetic system that doesn't need, sort of taking away the training wheels, doesn't really need DNA to hold its hand anymore. And then you could sort of try to recreate what we think happened, for the origin of life on this planet, which is that once you have that replicating molecule that can transmit information, you start to surround it in a very simple kind of cell, essentially a double layer of hydrophilic and hydrophobic molecules that either like or repel water. So you create a permeable membrane around the XNAs. And then that could be the basis of some sort of proto-life, a cell that is capable of making copies of itself.
So, in addition to this being absolutely thrilling for its own reasons, what are the advantages of a non-reliant on DNA form of life?
A lot of synthetic biologists have already tried to create small kinds of bacteria that can do useful things. For example, eating up oil that's been spilled in the ocean, decomposing trash, turning our wastewater into electricity. So the danger is that in tweaking native organisms, you might change their DNA in an unexpected way. And then if they start mixing with other organisms on this planet, you don't really know what kind of strange hybrids you're going to create, what sort of characteristics they're going to pass on to other bacteria that you might not necessarily want. So the advantage of XNA-based life forms is that they wouldn't really be able to do that, because the enzymes that we all have, that all life forms use to chop up DNA and RNA and work with them, they don't recognize XNAs. They can't do anything with XNAs. So if let's say a bacteria we created to clean up oil with XNAs escapes from the little secure area of ocean we want it to be in, it's not going to be able to breed with other bacteria or insert its XNA into other bacteria. It's going to be this sterile, ostracized, weird life form.
This speaks to one of the concerns that sort of the people who have concerns about GMOs talk about all the time. So this could be a remedy for that.
Yeah, that's an interesting point, because there has been a lot of research about how the wind very easily can carry with pollen from plants which are genetically modified. So this would sort of work. This would help with that as well, and that any pollen with XNAs wouldn't be able to fertilize organisms that have DNA.
But perfecting this process is still a long way off, correct?
All of the researchers I talked to were pretty clear that they don't see any applications like that probably until at least 10 years from now, maybe longer. But there are some more immediate applications. So for a long time, scientists have been tweaking DNA and RNA in subtler ways than what the researchers did with XNAs. What they do is instead of substituting an entirely new molecule in the backbone, they just change a bond or two in the backbone molecules. And this might make DNA or RNA a little more stable than usual. So XNAs turn out to be really, really stable because the enzymes in our own bodies, they don't recognize XNAs, so they don't break them down. So you could, for example, create short segments of XNAs that are designed to attach to pieces of DNA in our bodies or to proteins or other molecules in our bodies. And then you could inject these into somebody and they would last for a long time. They wouldn't be broken down in the blood. If you try to do that with DNA and RNA, they're not going to last as long at all.
Now, you mentioned 10 years, and that frightens me because 10 to 20 years is really the answer that everybody gives for every amazing technology.
That might be partially because, 10 years doesn't seem like it's that far off, right. But it's also not right around the corner. So it seems like sort of a good bet. I think it's really hard for anyone to come up with, a really precise prediction. What I will say, though, is that particularly when it comes to technology of working with genetics, we've seen huge leaps very, very quickly. You probably, you hear a lot about cheap genome sequencing. Mm-hmm. That's a technology that has become cheaper and faster and more efficient in leaps and bounds. It hasn't sort of hobbled along at all. So it's foreseeable that something similar could happen with synthetic biology. And certainly Craig Venter and others have been making pretty astounding achievements, even if nobody has yet created truly synthetic life that's 100% synthetic and artificial. They have been able to sort of create life forms from scratch, more or less, very simple cells, where they're piecing together the genetic material themselves, nucleotide by nucleotide.
I love optimists, sir. Ferris, thank you very much for being here.
You're welcome.
I'm talking to Emily Labor-Warren, the director of the Health and Science Reporting Program at the CUNY Graduate School of Journalism in New York City. Welcome, Emily.
Hi, thanks.
You wrote an entry for the World Changing Ideas article about some new research into Alzheimer's disease. So let's start by talking about what we do know about Alzheimer's. Do we know what causes it?
Nobody knows what causes it, and there are various contributing factors. The one that has been the most obvious is a protein called beta amyloid, which is basically when you look at someone's brain after death, someone who had Alzheimer's, what you see are these kind of hard little pellets of this amyloid protein that have built up all over the brain. There's something called the amyloid hypothesis, which suggests that that causes Alzheimer's, but more and more there are scientists who are starting to suggest that possibly amyloid is just a byproduct of other processes, and there are other things that are actually causing the disease.
And the reason why that's so tough to distinguish between whether it's a cause or an effect is because you can only see it by dissecting the brain, correct?
Well, until very recently, that was absolutely true. In fact, you really couldn't say for sure whether someone had Alzheimer's until after they died, because that was the only way. But there is a brain imaging technique called positron emission tomography. It's called PET, is how people refer to it, which is able to basically look at a living person's brain, and without harming them in any way, and see the amyloid deposits. So now it is possible to diagnose Alzheimer's earlier.
So tell us about the proposed research that's connected to this.
A lot of money, obviously, has gone into trying to create medications that would protect or cure Alzheimer's, because there's so many people who suffer from it. And they have all failed, every single one of them. Some people say they failed because they're just the wrong medication, maybe because a lot of them were geared towards amyloid, and maybe amyloid is not the problem. But there is a very passionate group of scientists who say, the problem is not that the medications are the wrong medications, it's that we've been giving them to people way too late, because what they learned when they started to be able to use the PET scans to see the brain of people who have Alzheimer's is that these amyloid deposits do not happen just when people are getting symptoms. They start to build up, 10, 15, 20 years before the person even starts to have any memory loss or cognitive problems. So what these scientists are saying is, we need to get to people before they even develop symptoms, because that's when their brain is already starting to deteriorate. And the problem is that we've been treating them too late.
So what's the research? How are we going to find that out?
There happens to be a family, an extended family in Colombia, who have the distinct misfortune of a very unusual kind of genetically predetermined Alzheimer's that it strikes earlier in their lives, so they tend to be seriously mentally disabled by their 50s. But the thing that is really very useful for scientists about this is that it's based on one single genetic mutation. You do a blood test. If you have it, you're going to get Alzheimer's in middle age. If you don't have it, you're not. So they're able to do blood tests of these people and find out, when they're still in their 20s and 30s, who is destined to get Alzheimer's and who isn't. So this is a perfect study population, because they chose a drug that's actually an anti-amyloid drug is the one they decided to use. They're going to give it to these people way before they develop symptoms. So basically when the amyloid is just beginning to accumulate, and see if that actually makes a big difference in their ability to prevent the deposits, and thus they are hoping in their hypothesis would be and prevent the progression of the disease.
So now this investigation involves 300 members of distantly related families?
Yes. The way they did it, they tried to be very, very sensitive to the concerns of these people. And basically, one of the things was they didn't want to just enroll people who had the gene that predetermined Alzheimer's, because that would mean some of the people did not want to know. They didn't want to know if they were going to get Alzheimer's or not. So what they're doing, they've divided this family into groups of 100 people, and they have one group of 100 people who are getting the medication, who have tested so that they're predestined to get Alzheimer's. Another group, also destined to get Alzheimer's, are getting a placebo. But then there's a third group of 100 people who are actually tested that they're not going to get the disease, and they're getting the placebo. So basically what that accomplishes is that no one in the trial knows whether they're predestined to get the disease or not.
There's also a group of Americans that are involved in this as well.
Yeah, there are a smaller number of Americans who, none of them have this particular genetic mutation where it's like you have the gene, you get Alzheimer's, but they do have other kinds of genetic mutations that make them predisposed to what's called early onset Alzheimer's, the kind that strikes in middle age as opposed to old age. And so they have culled a group of, I think, 24 or so Americans, and they're going to do the same thing with them. Basically, what they want to, of course, find out is, will they be able to extrapolate the results from this very sort of unique Colombian family to other populations of people who are predestined to get Alzheimer's.
So again, this is proposed research. It hasn't been approved yet. So what's that process?
It's very complicated. It's international research, and there are many bodies that need to approve it. And I know that the scientists involved were very sensitive, as I said, to trying to, not sort of be these Americans who come into this other country, basically, and say, oh, do this, that, and the other thing to you. And so they've really gone through all the processes here and in Colombia. And when I spoke with them for the article, they felt pretty confident they were going to get their approvals. They were very specific to say to me, we have not gotten approvals yet. And so we don't want to be talking out of line. We are still awaiting, the permissions to do this work.
So then even if the research is approved and the drug works, is that the cure for Alzheimer's?
Well, what's interesting is that, most people who get Alzheimer's don't get this early onset variety.
Right.
They get, Alzheimer's later in life. Most people also, they may have a genetic predisposition, they may not. There's so many different kinds of biological situations that lead to Alzheimer's, so that, it's not, I don't think anyone thinks it's going to just be this, if this works, no one will get Alzheimer's again. But I think that if this drug succeeds in this population in Colombia, it will definitely be very important, because it will confirm the amyloid hypothesis. The idea that amyloid plaques are the cause of Alzheimer's, and that if you can get to the amyloid before it builds up in the brain and flush it from the body so it doesn't build up, then people won't get Alzheimer's. That would be amazing. If they could learn that, they would really let scientists know exactly where to be focusing their efforts. But I think the scientists involved, they understand. They're not expecting for this to be like, this is it, it's the best drug in the world and it totally worked. What they do feel pretty confident about, and what they feel is really significant about their work, is that because they're able to get to this population so early, and they're going to do really detailed measurements of these people all along the way. I think it's a 5-year trial, and they're going to be measuring their cognitive abilities, looking at brain shrinkage, looking at amyloid, looking at another protein called tau. They have all these benchmarks. So they're going to be kind of tracking these people as their lives go on, as their Alzheimer's develops, or hopefully doesn't develop. And what they hope to provide to researchers for the future are really specific signposts, so that when other work gets done on Alzheimer's in the future, rather than, one of the problems with trying to make a drug against Alzheimer's is it takes so long to develop, so if you give it to people, you have to wait so long to see, well, did it work,
Right.
So they're trying to develop a very, very clear, specific sort of intermediate signposts that can, so in other words, like if they could say, well, we know that if tau protein goes up by this amount, that means the person is on their way to Alzheimer's. And if it doesn't, then they're not. And then future drug trials could just use that intermediary point and say, oh, look, the tau protein didn't go up. So we know this drug is having an effect. So they don't have to wait for the full-fledged disease. So they're really hoping that this trial will provide a lot of data that will be extremely useful to researchers in the future.
Emily, thanks very much for your time.
You're welcome. It was my pleasure.
You'll find these fascinating technologies and more in 10 World-Changing Ideas, the cover story from the December 2012 issue of Scientific American magazine. You'll find a link to Scientific American on our website at skepticallyspeaking.com. While you're there, click the links to Twitter, Facebook, and Google+, where you can stay up to date on show news, and to iTunes, where you can subscribe to the podcast, listen to past episodes, or leave a review. Thanks for listening, and we'll see you next week on Skeptically Speaking.
Bring it on, the skeptics await your letter. Your call, tweet, homing, pigeon, whatever. Desiree Shell gonna raise a little hell, metaphorically that is. 'Cause we're skeptically speaking.